Well, hello again. We're going to move away from proteins today and go back and take another look at carbohydrates. Now, Dr. Bartling talked about the structure of carbohydrates, and I talked about the metabolism of carbohydrates, particularly glucose, and how it's utilized for energy. But today we're going to take a look at some of the other functions that carbohydrates carry out. And we'll start by comparing and contrasting two groups of molecules, one which we call glycoproteins and the other proteoglycans. And as the names imply, glycoproteins are really proteins with some sugars attached to them, whereas proteoglycans have more sugar than protein, usually a protein backbone with much more frequent carbohydrate chains and longer ones as well. Now, before we get started, I just want to remind you we're finishing up week 4 here, moving on to week 5. Remember, week 6, which starts week after next, we have an exam on Tuesday afternoon. That would be the 29th, I believe. We will have a discussion session Monday morning to answer any questions that you might have. So want to start preparing for that. So let's talk about now these two types of molecules. And this is a diagram showing the basic idea. And on the left, on the far left, is a list of the number of the sugars or monosaccharides that get incorporated into these molecules. And you can see some of them might look somewhat familiar, particularly mannose, galactose, but other sugars are derivatives, and so they have been modified in some way, sometimes to put a nitrogen on them, an acetyl group on the nitrogen. Some of them are acidic, like sialic acid, but these are the main components of both of these types of molecules. Now, in the middle is a schematic of a glycoprotein. So generally these are globular proteins, can be enzymes, can be structural proteins that have some of these different carbohydrate complexes attached to them. On the right is an example or a schematic of a proteoglycan. And again, with these molecules, there's a polypeptide backbone with long linear chains, often very similar, if not identical to each other, that are branched off of the polypeptide chain backbone. Okay, so glycoproteins then contain short glycan chains, usually 3 to 15 sugars, sometimes a little more. They're highly branched, and that's as opposed to the proteoglycans. There's no repeating unit here, so I was going to say random, but it's not random, as we'll see. And they contain much smaller amounts of sugar than protein, usually maybe 10 to 15% carbohydrate by weight. Now, most cell surface proteins, secreted proteins, proteins found in the endoplasmic reticulum or in the Golgi are glycoproteins. And so you may see a trend here. Most of the proteins that cross a membrane, and that's usually into the endoplasmic reticulum or ER, and then it's trafficked out to another location. Most of these proteins are glycoproteins, so they are glycosylated after they pass through the membrane, whereas most cytosolic proteins don't have any kind of carbohydrate attached to them. And of course, if you look hard enough, you'll find some exceptions in either case. Now, the function of these glycoproteins, or the carbohydrates attached to the glycoproteins, is some of them assist in protein folding. So if you don't have the carbohydrate attached, the protein won't fold correctly and be able to function normally. They can also enhance protein solubility. As you might imagine, these sugars are highly soluble. So if we need the protein to be more soluble, we can attach some sugars to it to make it more hydrophilic. They can also stabilize the protein, and that's can be important as well, particularly those proteins that are presented on the outside of the cell or secreted. So the next point here sort of goes along the same lines, and they can protect the protein from proteolytic degradation. Another important role for these carbohydrates is to target the protein to specific subcellular localizations. We're not really going to talk about that today because we're going to spend some quality time talking about that in the near future. They can also serve as recognition signals for carbohydrate binding proteins such as lectins. And also, what our bodies or our immune system sees when it sees a bacteria is generally carbohydrate. And that is going to be much more immunogenic than the protein probably. So more often than not, if we are able to raise an antibody against a bacterium, potentially a pathogen, it will be directed at the carbohydrate on the surface rather than at a protein. Again, not always, but probably more often than not. Now again, these oligosaccharides on glycoproteins contain a variety of sugars. Some are the amino sugars, particularly N-acetylglucosamine and N-acetylgalactosamine. There's the neutral sugars, galactose, mannose, fucose as well. And the acidic sugar, sialic acid, also called N-acetylneurominic acid. Now, in contrast to the glycoproteins, proteoglycans contain as much as 50 to 60% sugars. And again, the sugars on these molecules are long, unbranched polymers. They may contain hundreds of monosaccharides, and usually there's a repeating disaccharide unit. So let's go back to the schematic diagram now that we've talked about these, and again we see in the middle the schematic of a glycoprotein. That's a protein. I'll show you an example in a little bit of a cell surface receptor. So they could be receptors, they could be enzymes, they could be other proteins carrying out other functions that have been decorated with these oligosaccharides. The proteoglycans, on the other hand, have this shorter polypeptide backbone, and then they have these long linear carbohydrate chains coming off of them. Okay, so the carbohydrates can be linked to amino acids in two different ways. And one we call the N-linked glycoproteins, the other the O-linked, because they're either attached to the nitrogen on the asparagine, shown on the left, or the hydroxyl group, in the case of the O-linked glycoproteins, of serine, and also the hydroxyl group on hydroxylysine. Now, you may not recall seeing hydroxylysine in the list of 20 amino acids that are incorporated into proteins. That's because it's not one that is. It is derived by a post-translational modification of a lysine. So the lysine gets incorporated into the protein. After that protein is made, then it's converted to hydroxylysine. And then it can be glycosylated. Not every hydroxylysine, of course, is glycosylated, but it does present another site for potential glycosylation. Also see glycosylation at threonine, which is very similar to serine, just has an extra methyl group. So it also has a hydroxyl group on the R chain. So here are some examples of linkages. And in each of the examples, the amino acid is shown sort of at the top in the right. I don't know if you can see this, but up here, and then the carbohydrate is attached in the figure A through an N linkage on the asparagine. B is through an O linkage on serine. In both new, in the top example, the N-acetylglucosamine is attached, and the second sample, N-acetylgalactosamine is attached. In the third example, C, there's a disaccharide attached to the hydroxylysine. And in the final example, another example of attaching a sugar to the hydroxyl group on serine. Now, when we look at these carbohydrates, we're seeing two types here in this figure. One that's often called the high mannose type, and the other one, I don't know if it really is called anything, but they label it tetraantinary. Not sure what to make of that, but they both have this common backbone with two galactosamines attached to the asparagine, and then mannose attached to that, and then two mannose branching off of that. So in the high mannose group, so again, this is the common linkage that begins the carbohydrate chains. So the high mannose group is also a precursor for other types of oligosaccharides. So after all these mannose residues are attached, they can be chewed back and other different sugars can be attached. Now, it says here that more than 100 different complex structures have been identified. That's probably a pretty low estimate. There's probably many more now. And when we look at these glycoproteins, we find that they could have a single N-linked oligosaccharide, or they may have several. If there are multiple ones, they could be identical or they may be different. Now, this last point that multiple different structures may be found at the same site on a glycoprotein, that means one protein molecule may have one structure and the second molecule of the same protein may have a different structure on it. That's generally considered to be due to incomplete processing, although there probably is some variation. Now, the typical structure for a O-linked uh, oligosaccharide is the attachment of N-acetylgalactosamine followed by a galactose and then a sialic acid. And that's shown here in both the schematic as well as the structure. Now, many proteins have both N-linked and O-linked structures on them. And again, the number of O-linked structures can vary from protein to protein. So here's an example of a glycoprotein. In this case, it's the receptor for LDL, or low density lipoprotein. People that don't have this receptor have a serious problem metabolizing cholesterol and generally die from heart attacks at a very early age. Even those people that have one good gene to make a viable receptor and one bad one, so they're heterozygous for the receptor, don't do well. They generally, again, die of heart attacks, usually in middle age or late middle age, so they're not as severely affected as somebody who has a defect of both genes. So this is the receptor that pulls cholesterol out of the bloodstream. And this is a schematic of its structure. It is embedded into the plasma membrane. At the end terminus is the part that binds to LDL, and attached to that are a couple of N-linked oligosaccharides. On the stem are some O-linked oligosaccharides. Now, these oligosaccharides are not involved in binding LDL. So once the receptor reaches the cell surface, we can strip the oligosaccharides off of the receptor and it still works just fine. But they do help in folding the protein in the endoplasmic reticulum. So without the addition of these saccharides, the receptor is not going to form properly and probably will not work quite properly, if at all. The O-link oligos on the stem are involved in stabilizing the protein and keeping it in its native state. So although these carbohydrates don't have a role in the function of the, direct role in the function of the protein, they do serve an important purpose. And again, in folding the protein into its active conformation and in stabilizing that protein. So if you disrupt that process, you're going to disrupt the metabolism of cholesterol, and you could cause some serious problems. Now, we touched on this the other day, and this just illustrates the point that normal cells can make pretty much any sugars from any other ones. The title of this is they can make it from glucose, and that's because glucose is a main monosaccharide that we ingest. But they can convert back and forth between glucose, mannose, galactose, or any other sugar. And this is a diagram showing some of those reactions. So again, we don't need to sit down and memorize those or belabor it, but just the important point is if you've got sugar, you can pretty much make any of the sugars that you need. The other point is that most of these intermediates involve either a phosphate group on the sugar, so they're kinase, or they're conjugated to a nucleotide. You see, that's quite common in these pathways to attach the substrate to a nucleotide before you start building the final molecule. Now this is a diagram showing the pathway from, in this case, fructose 6-phosphate, which is, of course, is an intermediary in glycolysis, to some of the other amino sugars and acidic sugars. And again, two things to remember. One is that we can make all these sugars from other sugars, and also um, it requires energy, and generally these sugars are attached to another molecule like a phosphate or a nucleotide in order to facilitate the movement through the pathway. So another group of carbohydrate-containing molecules that's gotten a lot of attention lately are the mucins. This is an example, a schematic example of a mucin. And again, this is a molecule with a core polypeptide and then a large amount of carbohydrate attached to it. In this case, showing the mucin attached to the cytoplasmic membrane of a cell through this anchor on the polypeptide. But not all mucins are attached to the cell. There are mucins in saliva, and there are mucins in other locations that are free-floating. So some of the properties of mucins. Now these are molecules that are secreted by epithelial cells that line the respiratory tract and gastrointestinal tract, the genitourinary tract. So any epithelial layer is likely secreting mucins. Again, these are very large molecules. Molecular weight can be over 1 million. The majority of that weight is due to the carbohydrate. And about 1/3 of the amino acids on the polypeptide backbone are either serine or threonine, and most of those are substituted with O-linked oligosaccharides. So there's a lot of sialic acid on these so that they repel each other and sort of spread the mucin out. So this forms a very viscous layer on epithelial cells or in liquids like saliva. So it's a protective barrier on the surface of these epithelial cells, also providing lubrication between surfaces. And it can also facilitate transfer. So it's a protective barrier, it's a lubricant, and if you don't have this, you're going to have some problems. And there are many, many different types of mucins, many different sizes and shapes. Again, some are anchored to the epithelial cells, some are secreted. There are a number of mucins found in the oral cavity. In fact, we in our research lab have initiated a study with a group at MIT who are experts on mucins to look at the effect of different moieties on the oral mucins and how they affect the composition of the oral microbiome. So there's some evidence to believe that, evidence showing that different people do have different types of mucins in their mouth, and there's some evidence to suggest that that may have an effect on what bacteria they have in their mouth as well. Now, where we find a good deal of, or quite a thick mucus layer is in the intestine, and this is a schematic of the intestine, a cross-section of the intestine, and you can, going from the bottom inside to the lumen of the intestine, and most importantly for this discussion, the top layer, this thick blanket of mucus or mucins. So when bacteria or other organisms enter the intestine, what they see is this layer of mucin. They don't see the cells beneath. And if you disrupt that mucus layer, then you can get invasion by bacteria into the tissues. So selectins are a group of proteins similar to lectins that are found in a lot of plants that bind carbohydrates. And this is an example here of E-selectin, which is found on endothelial cells, that is, the cells that line blood vessels. And this is used to pull leukocytes out of the bloodstream. So the carbohydrates on the leukocyte surface bind to the lectins, and that initially slows their movement through the vessel. Then it causes them to round up and eventually to penetrate between the endothelial cells into the surrounding tissues. And so when you have a signal, and you're going to hear much more about this in immunology, when you have a signal that something is wrong, the immune response can be initiated through this interaction between carbohydrates on the surface of leukocytes, or immune cells, and the binding proteins, in this case selectins, on the surface of the walls of the blood vessel. Okay, I just want to point out briefly a little bit of a difference between N-linked oligosaccharides and O-linked. This shows in terms of how they're made, this shows the synthesis of an N-linked oligosaccharide, and the way this is done is to anchor it on the inside of the endoplasmic reticulum, and once it's made, then it's transferred to the protein that has just come across the membrane of the ER. Now, once it is attached to a polypeptide, as it's moving through the membrane of the endoplasmic reticulum, it can still be processed. In other words, sugars can be added or cleaved off to form the mature saccharide. And that can happen as the protein travels through the ER and into the Golgi. Now, synthesis of the O-linked oligosaccharides occurs in the Golgi, and it is not anchored to the membrane, but it happens as proteins move through the Golgi, so a little bit different than the N-linked carbohydrates. Okay, so these oligosaccharides carry out a number of functions when they're attached to the proteins. They're involved in protein folding. So as we saw a minute ago, as the protein comes out of the ribosome and travels through the membrane of the ER, it can attach these oligosaccharides to it, and then that will facilitate folding of the protein into its proper conformation. These oligos can also be used to target proteins to specific sites in the cell. They also are involved in increasing the solubility as well as the stability of proteins. And again, as you can imagine, if we attach a bunch of very hydrophilic carbohydrate to a protein, it's going to make it much more soluble in water. And lastly, they can also be involved in recognition interactions, and I just showed you an example of leukocytes binding to epithelial cells. This is an example of targeting proteins to lysosomes by tagging them with these high mannose oligosaccharides. And we're going to talk about this more in detail when we talk about how proteins find their ultimate destination within the cell, or how they are transported out of the cell. I'm going to talk the last couple of minutes here about the surface of bacteria, starting with this molecule, which we generally call peptidoglycan. Another word shown here is muropeptide, although I can't recall anybody ever using that name. But basically, this is a long chain of carbohydrate. It's again a copolymer, which we'll look at in a minute. Cross-linked through these short polypeptides. And if we look at the surface of bacteria, and we can divide most bacteria into two groups, and I'm sure you've all heard of gram-positive and gram-negative bacteria. And this is what the surface of each of those types of bacteria look like. On the left are gram-positive bacteria. It's a little simpler structure. On the bottom, we see a typical lipid bilayer. And then on the outside of that is this thick layer of this peptidoglycan. And when you do a gram stain, this is what binds to the crystal violet. So basically, a positive gram stain means it binds crystal violet. And that's all it means. It doesn't mean anything about the function of the bacteria. Now, the gram-negative bacteria, the outer part of it is shown on the right here, and we start on the inside again with a typical lipid bilayer with proteins in it and spanning it or on it. That's followed by a very thin layer of peptidoglycan, very similar to the peptidoglycan on gram-positives, with one minor variation. And then on the outside of that is another lipid bilayer with this, on the outside of the lipid bilayer is this lipopolysaccharide, which I believe Dr. Barling has talked about. And I'm not going to talk about that today, but it's actually a pretty nasty compound. So both gram-positives and gram-negatives have peptidoglycan. The gram-positives have a thick layer on the outside of the cell or bacteria, whereas gram-negatives have a very thin layer between two lipid bilayers. So this is shown in another schematic here, showing on the bottom the inside of the, in this case, gram-positive bacteria. That's surrounded by the cytoplasmic membrane, which is a lipid bilayer, and again with proteins embedded into that bilayer or spanning it. And then on the outside of that, a thick layer of this cross-linked peptidoglycan. The gram-negative, again, starting at the bottom inside the cell, is surrounded again, just like gram-positives, by a lipid bilayer. Then there's a very thin sheet of peptidoglycan, and then outside of that is another lipid bilayer with the lipopolysaccharide on the outside surface of the bilayer. Okay, this is the repeating unit of peptidoglycan. There are two carbohydrate residues attached together, N-acetylglucosamine and N-acetylmuramic acid, lots of time called NAM, N-A-M. Attached to the NAM is a short polypeptide, 5 amino acids, and that's where these molecules are cross-linked. So if you notice here, the 5 amino acids, and when they're cross-linked, the last alanine is broken off, and this alanine forms a covalent bond with the lysine on another chain. So this shows it again. Now, the picture I showed you previously was from a gram-positive bacteria. This is from a gram-negative bacteria, and you notice it's the same thing, N-acetylglucosamine, N-acetylmuramic acid copolymer with a short polypeptide cross-linker. The only difference is this third amino acid away from the carbohydrate is a modification of the lysine. So that's the only difference. They form cross-links just the same as the gram-positive ones. It's interesting, though, in insects, the insect immune system tells the difference between gram-negative and gram-positive bacteria based on that variant in the amino acid at that position. It's not the case for humans, which recognizes the gram-negative bacteria by the lipopolysaccharide. Okay, so this then showing how these long chain copolymers of 2 sugar residues and then the amino acid short chains, the different colors representing the different amino acids cross-link to each other to form this tight matrix. This is showing an electron micrograph of a cross-section of a gram-positive. Remember, the gram-positive is the one with the thick layer of peptidoglycan. So you can see again at the bottom is inside the cell. There's a dark layer separating that from the peptidoglycan layer on the outside. Now I want to talk just a little bit about synthesis of peptidoglycan, and the reason is that, as we're going to see in just a second, this is a target for a number of antibiotics. And so this is a pretty detailed description of how this is synthesized. I don't want to go through it in detail, but just again, notice that this all starts by hooking the sugar onto a nucleotide, in this case UTP, well, UDP, because it loses a phosphate when you hook it on. And so this starts the process for the synthesis of these amino acids, and then as they're hooked on to the backbone carbohydrate. So let's look at a little less detailed diagram here. And what this is showing, starting up in the top, you start with N-acetylglucosamine. That gets converted to NAM, which is attached to UDP. Then the peptides are attached to the NAM. Then it's secreted out of the through the membrane, but held on by anchored into the membrane. At that point, then it can start to attach it to an existing chain. So you see at many of these steps along the way, there are antibiotics that prevent or inhibit that reaction. And these are generally naturally occurring antibiotics. They may have been derivatized to make them more powerful, but originally naturally occurring molecules that inhibit this process. So starting at the bottom, the last step in formation of mature peptidoglycan is to cross-link these chains, and penicillin inhibits that step. Now, this is why you have to have growing cells in order for penicillin to work. It really doesn't do anything on non-growing cells, because cells that aren't growing don't need to make any additional peptidoglycan. But if cells are growing and trying to divide, if you disrupt this process, it will break down will cause the peptidoglycan to not cross-link, and it will fall apart. Now, this is generally why penicillin is considered a good antibiotic against gram-positives, and you'll probably see in some places where they say penicillin doesn't work against gram-negative bacteria. However, common way of manipulating E. coli in the lab, which is a gram-negative bacteria, is to kill it off with penicillin. So in the lab, anyway, penicillin is very effective at killing E. coli, at least. Vancomycin inhibits the step prior to that, and that is the addition of this 2 sugar subunit onto the growing chain. There's a couple of antibiotics, which I really have never heard being used, that inhibit the first couple of steps in the process. So again, if we look at the gram-positive, cross-section of the gram-positive bacteria, we see a typical membrane lipid bilayer structure on the inside with a thick layer of peptidoglycan surrounding it. With the gram-negative bacteria, we see that same lipid bilayer on the inside, a very thin layer of peptidoglycan, and then outside of that is another lipid bilayer with lipopolysaccharide on the outside of that bilayer. Okay, some bacteria have additional layer of protection on the outside, this capsular material, which again is carbohydrate. This is showing a couple schematics of a couple of examples, gram-positive on the right, gram-negative on the left. And so you see, in addition to, on the gram-positive, in addition to the peptidoglycan, we see this thick capsular material shown a sort of yellowish color here. We see that same idea on gram-negatives. Now, some bacteria have a capsule and are encapsulated, others are not. In fact, some species of bacteria, some of the strains will be encapsulated and some won't. But if the bacteria is encapsulated by this carbohydrate, it can really serve to block phagocytosis, so make the bacteria much more difficult to clear in the body. It can aid in bacterial adhesion to host tissues, and it can also prevent drying out of the bacteria. So if the bacteria finds itself in a hostile environment, say it's an oral bacteria, and it finds itself lying on your toothbrush most of the day, if it's encapsulated by this carbohydrate material, it'll probably survive being in that kind of environment for a period of time until it finds itself back into a more hospitable environment. Now, different bacteria have different repeating units, and some are quite unique. I'm going to show you a couple of examples. This on the top is the bacterium Porphyromonas gingivalis, which is an oral bacterium that's associated with periodontal disease. And probably be hearing a lot more of it over the next few years. It's a pretty nasty bug. It um, synthesizes a large array of different um, protein or enzymes that can degrade proteins, um, and sometimes it puts them into a little membrane-bound sac and sends them out to destroy tissue. On the bottom is Pseudomonas aeruginosa, sometimes considered a soil bacteria, um, but found just about everywhere. Um, those of you who like to sit in the hot tub, this is the bacteria. If you're not careful, that tends to grow up in hot tubs, and you can have a culture that has billions of bacteria in an ml of hot tub water. I put this on here because the Pseudomonas actually has a fairly unique capsule, has unique properties. I'm not going to go into the details, but just it's a very, very interesting, unique bacteria. This is also the bacteria that causes problems with people with cystic fibrosis and tends to invade the airways, the upper lungs of people who are missing the ion transport channel that is the problem with cystic fibrosis patients. This is an example of another example of a capsule and a bacteria, in this case Bacillus anthracis. This is the bacteria that causes anthrax, and you can see the capsule shown in green here, why this might be a serious problem, because with a capsule like that, the bacteria can be pretty stable over a period of time. So again, if we look at a schematic, and this is the case of a gram-positive bacteria, we show from inside out the light blue represents the lipid bilayer, the cell membrane. Outside of that is the layer peptidoglycan, and then the light green layer surrounding that is the capsule. Here's another example of a bacteria, in this case a strep, and in this case it was stained with India ink. The bacteria takes up the India ink, but it doesn't bind to the capsule. So you can see in this case there's a tremendous amount of capsular material that is surrounding these bacteria. So the last thing I want to talk about is this extracellular polysaccharides found in another oral bacteria, in this case strep mutans. It's not the only strep that produces these kind of polymers, but it's the most common one. And of course, strep mutans is responsible for a lot of caries, major contributor to the caries process, because it, again, as we've talked about, it likes to chew up glucose and spit out lactic acid. Now, when it comes in contact with sugar, particularly sucrose, it will break that sucrose into fructose and glucose, and if it's hungry, or it will use some of that to go through glycolysis to generate the energy it needs. But if there's an excess, it will store them in the form of these polymers called fructans, which are polymers of fructose, and glucans, which are polymers of glucose. Okay, so this represents that, and we see the sucrose represented by a red and a black dot. Remember, it's the disaccharide containing one glucose molecule, one fructose molecule. So the first thing is to break those into monosaccharides, and then if the cell needs energy, it will take that glucose and run it through the glycolysis to produce energy. Take the fructose and generate these polymers, or fructans, that it can use later. So when it runs out of a source of food, it's got these fructans that it can break back down into fructose and then send those through glycolysis. Now the glucans are a little different. Not as likely to be digested. These are used to form an extracellular matrix around the bacteria. Okay, so when the strep mutans encounters a nice meal of sucrose, it can break the sucrose apart into fructose and glucose, use the fructose for energy, synthesize polymers from the glucose, which it can embed itself in and use those polymers or glucans as extracellular matrix. So I'll show you a couple of pictures of that. But this allows the strep mutans to stick to itself as well as to stick to other surfaces like the tooth surface. So in this picture, the cells, the strep mutant cells, are shown in black, and you can see this glucan matrix surrounding them. Show you another picture. In this case, the cells are pink, and all the yellow material is this extracellular matrix. So you can see this bacteria can form quite a bit of extracellular matrix, which makes it more difficult to disperse the bacteria, because it really helps it to stick together and to stick to the tooth surface. So just to summarize that, many streps, but not all, synthesize this extracellular polysaccharides by polymerizing both fructose and glucose, which they obtain from sucrose. This summary talks about the linkages. I don't think we're going to go into that because Dr. Bartling talked about that probably more than you wanted to hear. But again, remember that fructose polymers function as energy storage, and they can turn over rapidly in plaque if the bacteria needs sugar for energy. The glucans, on the other hand, can be used as an extracellular matrix to hold the strep mutant cells together, as well as to bind them to the tooth surface. Okay, that's it for complex carbohydrates. So we'll move on to another topic next time.